Hello, it's Mr. Matusiak, and I'm going to go over RNA and transcription with you in this lesson. Later on, we'll go over translation. So we're going to take a look at the structure of RNA, how it's different from DNA, and then we're going to take a look at the process of transcription, which is creating a copy of the DNA material, but in an RNA strand. And we're going to use that to create a protein through the process of translation. And all together, transcription and translation, we call this protein synthesis. So let's take a look at RNA. So RNA is using ribose nucleic acid instead of the deoxyribose nucleic acid that we see in DNA. So it uses ribose. It is single-stranded, where DNA is double-stranded, and it has uracil. So DNA had thymine, had the ATCG, and RNA has AUCG. So that is a couple differences there. And we can see these differences. We have the single strand here. Notice how we have R for ribose instead of the D's for deoxyribose. It's single-stranded, and there's that U, where instead in DNA we would have the T. So we have our U, our G, a C, and an A. There it is again there. There's the U. So the phosphate group is still there. The nitrogen base is still there, connected in the same way as we see in DNA. It's just the sugar is a little bit different. So let's take a look at different forms of RNA. There isn't just one type of RNA. There's actually three that we're going to take a look at, and they're pretty easy to remember. This one, messenger RNA, which we'll sometimes call mRNA, the M stands for messenger, it carries copies of instructions to the ribosome to make proteins. And so what we see here is that single strand right there. These are identified as uracil instead of our thymine. And this is done, and these are made in the nucleus, right? Because that's where the DNA is found. DNA is found in the nucleus of a cell. So messenger RNA is made from DNA. And so that makes sense for it to be made in the nucleus. Now, we also have ribosomal RNA, or rRNA, and ribosomes are made up of proteins and ribosomal RNA. So as we see here, this whole structure here is a ribosome, and what we see in here is the ribosomal RNA. So all we really need to know for our class is that a ribosome is a protein with rRNA. That's all you really need to know about rRNA. Now we also have tRNA. The T stands for transfer. And the transfer RNA will transfer an amino acid. So this little guy up here is an amino acid. And it's going to transfer that amino acid to the ribosome. So a lot of stuff's happening in the ribosome. Remember, ribosomes build proteins. And amino acids are the monomers of proteins. And so this transfer RNA will come into the ribosome and drop off this amino acid. And inside of the ribosome, the amino acids will be bonded together to make a long chain, a polypeptide chain, which is a polymer. And that polypeptide chain will later on become a protein. So let's do a quick vocab check here. RNA molecules, they are produced by copying part of that nucleotide sequence of DNA into complementary sequence in RNA. So we unzip that DNA strand, we look at one side of that DNA strand, and we make a copy of it, but in RNA form, and that's called transcription. Now, this is all done by RNA polymerase. Remember, when we did DNA replication, we used DNA polymerase. Well, now we're making RNA, so we use RNA polymerase. So here's an overview, the central dogma, or the central idea. We're going to go from DNA, and we're going to make mRNA through transcription, and that mRNA will go to a ribosome and be translated or read, right? When you translate something, you're reading it and figuring out what it means. And when we read that information, which is how to build a protein, well, then we can actually build it. So let's take a few looks at the steps of transcription. Remember, it's completed in the nucleus. That RNA polymerase is going to attach that DNA molecule. It's going to attach at a point called a start signal. And we'll take a look at what these start signals are here later on. But that helps it figure out where to attach. So that DNA strand is separated, right? And we only use one side to serve as that template. Next, the RNA bases, the A's, the U's, the C's, and the G's, will attach to that complementary DNA template that we're using, that side of the DNA molecule that we're using there. And messenger RNA is made. And so then the mRNA, when finally it's done being made, it will detach, it'll leave the nucleus, and it'll exit the nucleus into the cytoplasm of the cell and find a ribosome, because that's where proteins are made. And we need to send the information, the instructions to build a protein, which is the messenger RNA, to the factory to build it, which is the ribosomes. Here's a picture of said mRNA being transcribed there. We see that we have our RNA polymerase. We have unwound a section of the DNA. We're just reading this one side. So this is the template strand right here. And we're attaching different RNA bases and creating a RNA strand, single-stranded, and this is our messenger RNA. So the language of mRNA, right? We use those 4 letters, the A's, the U's, the C's, and the G's, and the order that they come in means something, right? So we read them 3 letters at a time, and that's called a codon. And AUG, AAC, these are both examples of codons. So 3 nucleotides at a time. And each one of those codons will specify a particular amino acid. So a certain amino acid. So that AUG, it stands for a certain amino acid. Same with the AAC. All right, so just like I said there, each codon specifies a particular amino acid, and that's going to go in order in a polypeptide chain. The order matters. Now, some amino acids can actually be specified by more than one codon. All right, so what we have here is the wheel of codon. And how you use this is you start in the middle and you work your way out. For example, one of those codons was AUG. Let's see what that does. AUG, methionine. There we go. So this is an amino acid right here. All of these on the outside are different amino acids. And notice how AUA and AUC and AUU all code for this isoleucine amino acid. So that's what I meant by some amino acids can be coded by more than just one codon. So AUG is the code for methionine. CAU is the code for histidine. Let's practice that one. Start in the middle. CAU, histidine. And UCU is the code for serine. So UCU. There we go. Notice there's also a stop. So UAA, UAG, those code for stop. So when a ribosome reads that, it knows it's done making the polypeptide chain, it's done reading the mRNA strand, and it's time to detach that polypeptide chain. So just like I said there, AUG is our start codon. All right, it's methionine, but it can also be our start codon. So you'll see all of our examples that we're going to use, they're going to start with AUG. And then there are 3 stop codons, which I just showed you there. All right, so real quick, break down what we were just talking about. We take that DNA, we unwind it, right? We use this template strand, and we make our RNA. Check it out, AUG, start codon. And then we're going to use that transcribed RNA, and we're going to translate it. We're going to get more into translation later, but I just want to give you a quick rundown on transcription and how we use it. All right, so we'll come back next class. We'll put these ideas to work, we'll model it, and I'll answer any questions you have then.